Bisphenol A, or commonly known as BPA, is a carbon-based synthetic compound that has become an important health concern in recent years. Most people are unaware that BPA is pretty much everywhere. Your reusable plastic container,
beverage can liners,
food wrap,
plastic bags,
baby bottles,
toilet paper,
dental sealants,
and more. See, the problem is BPA can leach from these products into food or beverage contents, saliva, or even dust particles. A major route of exposure, therefore, takes place by ingestion or inhalation. Although BPA has a half-life of 4 to 6 hours, it has been found to persist in physiological systems for up to several days, and once it enters the body, it does quite a bit of damage.
Today, we'll be discussing how BPA exposure impairs reproductive function by disrupting biochemical pathways such as steroidogenesis, the production of steroids such as sex hormones.
Endocrine disrupting chemicals, or EDCs, are exogenous agents that interfere with biological processes regulated by endogenous hormones.
Among the EDCs, BPA belongs to the family of diphenylmethane derivatives and bisphenols, all of which are marked by the presence of two hydroxyphenyl groups.
BPA has been shown to disrupt steroidogenesis in animal models by decreasing the expression of key enzymes in this pathway, resulting in impaired reproductive function.
Ovarian steroidogenesis requires cooperative interactions between theca and granulosa cells, which are two cell types in the ovarian follicle.
Theca cells respond to luteinizing hormones, or LH signaling, by increasing expression of enzymes necessary for conversion of cholesterol to androgens.
Granulosa cells respond to follicle-stimulating hormone, or FSH signaling, by increasing expression of enzymes necessary for conversion of theca cell-made androgens into estrogens.
BPA has been shown to alter this pathway by decreasing mRNA expressions of 4 key steroidogenic enzymes.
STAR and P450-SCC catalyze the rate-limiting step, the conversion of cholesterol to pregnenolone, and therefore control the overall rate of the steroidogenesis pathway. We will now spend some time focusing on the structures and functions of these enzymes, as well as the biochemical implications of their exposure to BPA.
Steroidogenic acute regulatory protein, or STAR, is a transport enzyme that regulates cholesterol transfer within mitochondria. It is a 285 amino acid protein with 4 chains. Only its C-terminal alpha helices, highlighted in yellow, interact with the outer mitochondrial membrane. Each chain consists of a catalytically active STAR domain, which forms a pocket that binds cholesterol molecules for delivery to the enzyme P450-SCC.
P450-SCC, or cholesterol side chain cleavage enzyme, is a mitochondrial enzyme that catalyzes the conversion of cholesterol to pregnenolone. The heme group, shown in red, is the site of catalysis. The ligand, 22-hydroxycholesterol, shown in white, occupies 2/3 of the active site. Cholesterol is the steroid precursor for all sex hormones in the steroidogenesis pathway.
In the theca cells, STAR is a protein that regulates the transport of cholesterol from lipid droplets in the cytosol to the inner matrix of mitochondria.
This is the rate-limiting step of ovarian steroidogenesis. Without this transport, cholesterol cannot be converted to pregnenolone by P450-SCC, which is localized in the mitochondria. P450-SCC is a mitochondrial enzyme in steroidogenic theca cells of the ovary that catalyzes the conversion of cholesterol to pregnenolone via 3 monooxygenase reactions. This involves 2 hydroxylations of the cholesterol side chain, which generate 2 intermediates. The final step cleaves the bond between carbons 20 and 22, resulting in the production of pregnenolone and icosapro-aldehyde. Each monooxygenase step requires 2 electrons from NADPH. The electrons are transferred from NADPH to P450-SCC via 2 electron transfer proteins, adrenodoxin reductase and adrenodoxin. These 3 proteins constitute the cholesterol side chain cleavage complex.
BPA may downregulate STAR and P450-SCC production by decreasing mRNA expression of one or more of their nuclear receptors, or transcription factors that bind to DNA to regulate expression of target genes, in this case STAR and P450-SCC genes. Steroidogenic factor 1, or SF1, is a nuclear receptor that promotes the transcription of STAR and P450-SCC genes in the ovary. Upon LH binding to the theca cell receptor, a CAMP-dependent phosphorylation cascade acts as a signaling pathway to trigger SF1's activity. One hypothesis is that BPA can modulate SF1's activity by disrupting its active expression so it cannot promote transcription of STAR and P450 genes.
SF1 is only one possible transcription factor in ovarian steroidogenesis that may be a target for BPA. Current research has identified other possible factors, including those involved in negative transcriptional regulation. However, more research is needed to isolate an exact mechanism for BPA's regulation.
Due to rising public health concerns, BPA has been widely replaced by Bisphenol S in many BPA-free consumer products. However, preliminary studies suggest that BPS may also have endocrine disrupting effects in humans, such as a reduction of basal testosterone secretion. Thus, more research is needed to further study the pathways of human exposure to BPA in order to identify its potential health consequences, as well as safe alternative substitutions.